The isolation, characterization, and identification of Bacillus barbaricus by Aubrey Carlson. The purpose of this project was to discover if the types of bacteria in the area could be candidates for bioleaching applications. Bioleaching is a biotechnological process where microorganisms extract different metals from ores, minerals, or deposits. Microbes that are good candidates for bioleaching are capable of metabolizing specific metal-containing compounds. This project has the potential to expand the usefulness of microbes in many different ways, specifically bioleaching. The sample location was Mineral Mountain in the upper audit, bottom of the shaft at the east wall. The GPS coordinates are as follows. This location was chosen because this area is rich in solubilizable metals and the bacteria living there may have adapted in utilizing these metals. To determine what the colony looked like, I streaked the bacteria on a nutrient agar plate. As you can see from the picture, the colony was a small white opaque oval at about 2 centimeters in diameter. To determine the oxygen requirements of the bacteria, I inoculated an agar deep. There was growth down the agar and on top of the agar, as you can see from the picture. And this indicates that this bacteria was a facultative anaerobe. I wanted to determine the morphology of the bacteria, so I performed a negative stain at 1000 magnification. The shape and arrangement of the bacteria is Streptobacilli, and it ranged from about 2 to 10 micrometers in length. To determine the gram status of the bacteria, I performed a gram stain, and I viewed it under 1000 magnification. From the picture, you can see that most of the bacteria are pink, but there are some purple bacteria. This is considered gram variable or gram positive. I wanted to know the catalase status, so I performed a catalase test. Bubbles formed immediately, indicating a positive result for catalase. To determine the result of an oxidase test, I performed an oxidase test. The filter paper did not change within 30 seconds, so it was determined to be oxidase negative. To determine the motility of the bacteria, I performed a hanging drop motility test. The bacteria were spinning around at random and not uniform, so they are perceived to be non-motile. To determine if this bacteria could reduce nitrate, I performed a nitrate reduction test. When I added the first reagents, no color change happened, as you can see from the first picture. I then added zinc to the bacteria and it turned red. And this indicates that the bacteria was negative for nitrate reduction. I wanted to know if this bacteria had the ability to form endospores, so I performed an endospore stain at 1000 magnification. There are a lot of bacteria with green endospores, and this indicates the bacteria does form endospores. I wanted to know if this bacteria could detect iodine, so I prepared an iodine detection plate. The colonies turned a white color, as you can see from the second picture, and this indicates there's no trace of iodine. I wanted to know if the bacteria could detect any cyanide, so I prepared a cyanide detection plate. The colonies turned into a light brown, as you can see from the second picture, and this indicates that this bacteria cannot detect cyanide. One important part of this project was to discover if this bacteria could solubilize metal. So I performed a metal solubilization test for gold and silver. For the gold, no change happened after incubation with ascorbic acid. This indicates the gold solubilization was not detected. For the silver, the copper wire had some plating on it, so this indicates that silver solubilization was detected. To isolate the DNA of the bacteria to prepare for PCR, I centrifuged my culture and added a buffer to start cell lysis. The cloudy mixture indicates that cell lysis has been achieved. Polymerase chain reaction. The purpose of this is to replicate a region of the bacterial chromosome, specifically the 16S small subunit rRNA, to prepare for DNA sequencing. To view the results of my PCR, I prepared my sample and placed it into a gel electrophoresis. The electrophoresis was successful and the bacteria was about 1,500 base pairs. The 16S small subunit rRNA sequence was obtained and I analyzed this information in a program, and I obtained my clean trace data, as you can see from the first picture. I then blasted it to find close relatives. And the second picture is the top alignment match with the query and the subject. This is a table of my isolate compared with a dichotomous key and my top two results of my DNA sequencing. Since my colony matches Bacillus barbaricus in all parameters, it is Bacillus barbaricus. Based on a comprehensive set of morphological, physiological, biochemical, and molecular characteristics, the bacteria isolate is most likely Bacillus barbaricus. The results of the tests ran of the isolate match the profile of Bacillus barbaricus. This is the most accurate identification for the isolate in this study. The ecological roles of Bacillus barbaricus is that it promotes plant growth. It also has biocontrol of pathogens and pests. It is most commonly found in soil, dust, and water, and this bacteria are ubiquitous in nature. It makes sense that we would find this bacteria in the location of our isolate because they are known to be found in soil and water. They are also ubiquitous, meaning they are widely distributed and are found in many places. Thank you. The end.